Now that we know how substances can diffuse across membranes, let's explore osmosis in more detail. You know it is the movement of water across membranes, and most osmosis occurs through aquaporins. But will the water flow into the cell or out of it? That depends. Tonicity is the comparison of solutions to determine the direction of water flow across a membrane. Tonicity is relative, as we must be comparing two solutions. For example, the solute concentration of a solution inside a cell to the solute concentration of a solution outside a cell. We are assuming that the solute cannot diffuse, so only the water can flow across the membrane. The three types of tonicity are isotonic, hypotonic, and hypertonic. Remember, we have to be comparing two solutions to use these terms. We will examine the three different types of tonicity by using a red blood cell. We are assuming the membrane is not permeable to the solutes we are investigating, so we are only looking at osmosis, the diffusion of water. Isotonic solutions have equal solute concentrations. If a red blood cell is placed in an isotonic solution, there is a dynamic equilibrium. Water molecules still move in and out of the cell, but there is no net movement of water, meaning the same amount of water that moves out will move in. In isotonic solutions, the solute concentration outside the cell is the same as the solute concentration inside the cell. This is a red blood cell in an isotonic solution. It normally has that depression in the center. We say that the red blood cell is in osmotic equilibrium in isotonic solutions. A hypertonic solution has a greater solute concentration when compared to another solution. In this case, there would be a greater net movement of water out of the cell. This is because only the water can diffuse, not the solutes. When a cell is in a hypertonic solution, there is a greater concentration of water inside the cell. As there is more water inside the cell, it moves outside the cell by simple diffusion. Here's a good way to remember it. Water always moves to the area of greater solute concentration. So the red blood cell in a hypertonic solution would lose water and crenate or shrivel. A hypotonic solution has a lower solute concentration compared to another solution. If a red blood cell is placed into a hypotonic solution, there will be a net movement of water into the cell. As there is a higher water concentration outside the cell, the water moves into the cell by simple diffusion. Again, the water is moving to an area of higher solute concentration. A red blood cell in a hypotonic solution will swell and potentially burst if it takes in too much water. But what about plant cells? The cells of plants are surrounded by a tough cell wall, and while osmosis occurs, it is somewhat altered. This healthy-looking plant has cells that are in hypotonic solutions. There is enough water flowing into the cell to keep the water pressure, also called osmotic pressure, high enough so that the water pushes against the cell wall. This keeps the cell turgid, like an inflated balloon. But this plant is in a hypertonic solution. Water has moved out of the cell, and it is not turgid anymore. There is no longer enough osmotic pressure inside the cell, and the membrane detaches from the cell wall. The whole cell takes on a deflated look, and the cell has plasmolized. So far, we have only discussed the movement of substances by simple diffusion in cells. However, energy can be used to actively transport atoms and molecules. Energy must be used in active transport, as substances are moving against the concentration gradient. In other words, solutes are moving from an area of lesser concentration to an area of greater concentration. In most cases, the energy-carrying molecule ATP will provide the energy for active transport to occur. The sodium-potassium pump is an example of active transport utilized by most eukaryotic cells. It is called a pump because it actively pumps substances across the plasma membrane. The sodium-potassium pump is a carrier protein. In its inactive conformation, It is open to the interior of the cell. 3 sodium ions in the cytoplasm of the cell are attracted to specific regions of the pump. With the binding of the sodium ions to the pump, ATP is called into action. The ATP molecule hydrolyzes and transfers a phosphate group to the pump. This is known as phosphorylation. When ATP phosphorylates a molecule, that phosphate houses a high level of usable energy. The binding of the phosphate results in a conformational change in which the pump changes shape. When it changes shape, it opens to the exterior of the cell and releases sodium ions. And when it is open to the outside of the cell, 2 potassium ions are attracted to the pump. As the potassium ions bind, the phosphate detaches, and the pump returns to its normal conformation, releasing the potassium ions into the interior of the cell.